syllabus. So pretty easy. We have a post-quiz on Friday, and then next week on Wednesday is what? Exam number 1. All right, so how are we going to study for the exam? Watch the lectures. Not a bad idea. Not a bad idea to review quizzes. I don't anticipate I'm going to recycle any specific quiz on purpose or quiz question on purpose, but they're nicely comprehensive. So they're open after the quiz closes. So you just go to your grade book and click on your score, and then it should pull up the question and the answer, or answers. Other one is your lecture notes. So some of you have been good about coming into my office and asking questions about your lecture notes, and kind of just dialogue back and forth a little bit about what you've written down, what we talked about, maybe what I meant by certain slides, or what I wanted you to take away. Okay? 50 questions. You'll have the full class period. We won't have a lecture afterwards. There's no other assignments next week. There's not a pre-quiz on Monday. Okay? But we've got the immune system to at least introduce. We're not going to get through everything, as you can see. We've got today, and then we've got Monday on the immune system. And then after exam 1, we'll come back together and we'll talk about hypersensitivity. We'll talk about some of the immune disorders. Okay? But we're going to be setting the stage for discussing the immune system before the exam. So this content today and Monday will be on the exam. Okay? 50 questions, multiple choice. There'll be some questions on the screen. Images, either gross images, histology possibly, stuff you're familiar with. Any questions on the exam? Okay, well that's good. You guys are all set? You ready to dive into immunology? There's Richard. I was waiting for you, my friend. We were stalling for Richard. Okay. First up, what do you call a cat that lacks, or sorry, what do you call a cat that likes lemons? A sourpuss. Very well done. Comes late but starts participating right away. I like it. What kind of bird can lift heavy objects? A crane. Okay. Why don't cheetahs like to take baths? Why do cheetahs not like to take baths? They don't want to be spotless. All right. So we're in this section now, which is called the specific or adaptive immune system. We kind of have two names for it. The specific or the adaptive immune system. And the picture on the screen is a scanning electron micrograph of a lymphocyte. So what I want to do to start out is I actually want to back up a tad and talk a little bit about some review of what we've already covered. And then we'll get into the specific or the adaptive immune system. So remind us, what was our first line of defense? Barriers. And our second line of defense? Inflammation. And so now what we have is the adaptive immune system or the specific immune system, and we call this our third line of defense. Okay, and the book characterizes it that way. That's fairly standard nomenclature. And so if we look hierarchical, and we look at the immune system, we really can break it down into two branches. We can call the first branch our non-specific system. And then we have what we're going to talk about now, which is our specific immune system. Okay? And then under our non-specific immune system, we had really two branches that was first line of defense, which were barriers. And then our second line of defense, which is what? Inflammation. Okay? And over here we have specific or adaptive. And let's talk about these nomenclatures, why we characterize it this way. Well, non-specific is the same every time. Your skin doesn't change depending upon the invader, which actually would be kind of a cool sci-fi movie, right? If your surface of your skin could kind of adapt quickly to what it's experiencing, that would be kind of cool, I think. Right? You guys can borrow that if you read a movie, okay? Just put me in the credits. Non-specific inflammation, same every time. You get a cut today, you get a cut tomorrow, you get a splinter today, you get a splinter tomorrow, or your neighbor, it always kind of looks the same. We've got our first responders, our neutrophils, we've got our second responder, our macrophages, right? And so we've got characters in this scene over here on inflammation where we've got our neutrophils, right? And then we have our macrophages, our tissue macrophages that come from our monocytes. And these guys are referred to as white blood cells out of the bloodstream. And another specific name for them are what? Leukocytes. You guys with me? This is a review, so hopefully you're not like, we talked about that? Okay, so now we're going to move away from non-specific, which is the same every time. We're going to move over to specific or adaptive because this does, in fact, customize to what the invader is, which is kind of cool when you think about that. So our body has first, second, third line of defense. And our third line of defense over here is our adaptive immune system. And these cells are called lymphocytes. Uniquely different than leukocytes or white blood cells, these are lymphocytes. And these lymphocytes come in two different variations. So we have what we call B cells, or B lymphocytes, and we call the second type T cells, or T lymphocytes. And I'm going to flash forward. Maturation is why the tissue that they mature in, we'll talk a lot about this as time goes on, is where they get YB, YT. So these guys mature in the bone marrow. So they're B cells. And these guys mature in the thymus. They mature in those tissues. Become functional in those tissues. They grow up in those tissues. Make sense? This system here changes as you age. This system here on the adaptive, this is the whole functionality or mechanism by which vaccinations take their hold on us or have effectivity, is because of our adaptive immune response. And we'll talk about that and that'll become more clear. So immunizations are over on the adaptive side. It's a learned immune system. It changes as you get older. It matures. It has a memory. It learns. I mean, arguably you could say that it evolves. It absolutely evolves. So from the very beginning of when we're born, we actually, if we breastfeed as a newborn, we actually get some antibodies that are manufactured by these B cells. Okay, so these guys make antibodies. And these guys are what lead to cytotoxic activity. Ooh, that was strong. Okay. Didn't know if that was a permanent marker or not, but it's like, if I start waddling up here, just let me know. Okay. So antibodies come from B cells. Cytotoxic activity, killing a cell that's infected, it comes from the T cell. The other thing to keep in mind on the antibodies is these are usually humoral. This means in the blood. This word means in the blood. Okay? Circulating around. And these cells could be in the bloodstream or they could be in tissue. Make sense? So now you're looking at this outline of complexity of our immune system. One could argue that there's a lot of overlap. There's some redundancy built into this. What does that tell you immediately when you hear that terminology or you see that scenario that there's redundancy in this system? It's actually really important. And after the exam, we'll talk about where some of this stuff fails and why we have certain immune diseases, okay? But first, before we get there, we have to talk about normally what's happening. So I need you to really have a broad, comprehensive understanding of everything that's going on before we start talking about HIV, for example, right? You guys following? Okay, any questions on this? This is kind of an important first step for us to get started. I mean, this is the title slide, so we haven't even gone anywhere yet. We good? All right, so let's get into some of the details for today. So very quickly, with this listed out here on the board, which is going to be sort of our roadmap or our guideline of where we're headed for today and Monday, we're going to start walking through a lot of these details. And so if you haven't yet read the chapter readings or the pages that are assigned, I'd encourage you to do that. I think it'll be repetition and helpful to hear it a couple of different times. This material tends to be rather complex for students. So this section and the cancer section seem to be two kind of speed bumps for us in the semester, where it's kind of like we got to slow down a little bit. And some of you might see, well, he's not moving nearly as fast as he usually does. That's intentional. Like there's a smaller slide deck for today and Monday, yeah, because we're not going to go that quick. Okay? So you can see real quickly we get all the way into the detail fast. So we're characterizing on this slide, we've got the humoral immunity, which is another name for antibody side of things, talking about B cell activity, talking about these lymphocytes known as the B lymphocytes that are part of the specific adaptive system, our third line of defense. So right here, starting out, we just go, right? We're down in the details already. And so that's why I wanted to put this up here and write it out so we go slow, you guys craft it, and it gives you a perspective of where we were and now what we're about to talk about. And so you can see here, humoral, there's a bacteria that is seen. The B cell, labeled with the nice convenient B, is secreting antibodies. These antibodies bind and neutralize this bacteria. And when they do that, they actually get phagocytosed by the macrophage, and then the macrophage actually destroys it. So wait a second, are you telling me that these guys, these tissue macrophages, have a role over here? Yeah, absolutely. They just came over and helped out these B cells. They were manufacturing these antibodies. So the immune system that's specific is the B cells are producing these antibodies, they're labeling something, and then telling the second line of defense, go clean up this mess. Okay? So they're talking to each other. Now over here on this side, we have what we call cellular immunity or cytotoxic activity. This process looks a little different. So now we've got the virus that's infected, for example, this particular cell. This is a host cell. This is one of your cells. This happens to be what we call an antigen-presenting cell because what it does is it takes a component of that virus and it displays it on the cell surface. And by displaying it on the cell surface, it presents the antigen to the T lymphocyte. This T lymphocyte, known as a helper T cell in this particular example, releases cytokines, and that causes cytotoxic T cells, which are the effector cells, to group, and then they go and they actually kill the cell that's infected, and they, in this case, would target any phagocytic cell that actually has that infection displayed. Okay? You guys follow? So, on specific immunity, we've got kind of two different types of mediation. We've got what we call CD4 and CD8. So what the heck is a CD? Okay? Because my parents talk about it, and they still have them, but we just use Spotify. So, different kind of CD. Here's a cell. There's going to be some display molecule on the surface, okay? And it stands for a cluster of differentiation, otherwise abbreviated CD. And so these are markers. These are cell markers that are like little labels. Stem cells have it. All cells have markers on their surface that identify what they are. So if we go back to the last lecture, we talked about the regenerative fluid technology that was coming from mesenchymal stem cells. Those stem cells have CDs on their surface. That's how we know that they're mesenchymal stem cells. But they're markers like CD90 and CD105. These clusters of differentiation are called either CD4 or CD8. So CD4 is expressed on 60% of mature T cells. So 60% of these cells are CD4 positive. Okay? 40% of these T cells are CD8. Okay? The cells that are CD4 positive have a name associated with them. These are actually referred to as helper Ts. And the CD8s are referred to as cytotoxic Ts. Okay? You guys with me? So the CD4 positive T cell you see up here, that's the helper T cell. That's what you're seeing in this picture, this diagram. So we'll go CD4, this is a helper. Should you know these delineations? Yes. Just memorize them. Helper T cells are CD4 positive. Cytotoxic T cells are CD8 positive. They are two distinct populations of cells. And you can see that the helper T cells outnumber the cytotoxic T cells. Okay? I'm going to flash forward, just give you an insight. The helper T cell is the problem associated with HIV. So HIV actually depletes your helper T cells. They're called helpers because they help facilitate an immune response. And we'll talk about co-stimulation and activation of T cells and you'll see the role that they play. But you can kind of keep in the back of your head, okay, so this is the one that HIV virus infects. Okay, and then it progresses from there. HIV progresses into an acquired immunodeficiency disease or syndrome, AIDS. Okay, so this is where the problem is with HIV. Okay, so we got CD8, we got CD4, and then our antibody mediated, we have B cells, plasma cells, and we've got antigen presenting cells. So here, we'll go in this direction. We have B cells, we have plasma cells, and then we have APCs under the antibody mediator. All right, so let's talk some unique characteristics. So my drawing is much more simplistic on this CD, but you can see up here how complicated this interaction gets. So this top cell, this is this phospholipid membrane. This is an antigen presenting cell. It's presenting to a cell that is expressing CD4. So this down here, by definition, if it's expressing CD4, is what type of cell? Helper T cell. So now you've got an antigen presenting cell presenting this peptide, like it's like, oh, look what I have, right? And it's presenting this antigen peptide to this helper T cell. You know it's a helper T cell because it has this label that says, I'm a helper T cell, CD4. Well, you can appreciate how it's binding in a complex fashion. The CD4 is important because it interacts with this MHC class II molecule. Okay, so let's draw that. So here's a helper T cell, right? It's going to see an antigen presenting cell, which is an APC, and this one communicates with MHC class II. You guys with me? What do you think the CD8 cells communicate via? MHC class I. We'll get there, but okay? Perfect. That's where we're headed with all this. So you can see here, we had MHC class II, there's an alpha and a beta chain. That's that blue structure here. That bonds the molecule that it's presenting. It gobbled this up. And it's displaying it, and it's displaying it to the helper T cell because that's what binds with the MHC class II. And this triggers to the nucleus of this helper T cell signal number 1, saying, look, I found something. You probably need to do something about it. Well, there's actually a safety mechanism, and that safety mechanism is what we call co-stimulation, or specificity due to a co-stimulation event, which is signal number 2. So on this helper T cell, you have another CD molecule known as 28, and it interacts with CD80 or 86 of the APC, and that interaction is signal number 2. In order for this guy to actually be activated, it needs to see both signals. So you guys may not remember these movies, but back like in the 80s, there were always these movies where we were in the Cold War, and instead of just having the launch sequence for a nuclear missile to Russia, you would actually have to put in 2 separate keys by 2 different individuals, and they would go like 3, 2, 1, and they would turn at the same time, and then the launch light would light up. And so that's how I think of this. Okay, I think you can see some, representations of that idea in some of the movies today. But this is sort of your body's way of making sure that if you accidentally dock an APC with the helper T cell, and the CD4 sees the MHC2, but there's nothing being presented, then you don't get automatic activation of the helper T cell. Okay? So the CD80 and 86 only gets co-localized to the membrane when it's displaying an antigen. So let's draw that. So here, we've got something else. We've got cluster 80 or 86, right? Either one. And it's going to interact with, what is the co-stimulatory molecule? CD what? 28. You see? You guys following? Okay, so now we've got this particular signal is signal 1, and this particular signal is signal 2. You guys follow? And this sequence is called co-stimulation. That's that terminology. So what we say is, this process with the helper T cell and the antigen presenting cell has tremendous specificity, because you've got very specific molecules that are involved in order to send the signal. We're going to get to it a little bit later, but it also has memory. So when it sees that antigen, that yellow ball, that sphere that it's presenting here, later on down the line, that helper T cell will create what we call memory T cells. Okay? And they will remember this antigen in the future, and then the second response is faster and more aggressive. And we'll get there. Okay, now what's going on over here with panel B? This is a similar kind of situation with co-stimulation, but instead of involving the helper T cell, this is actually a B cell. Okay, so that's this particular one that matures in the bone marrow and makes antibodies. So similarly, we've got a signal 1, signal 2 event that takes place. We've got an antigen that binds. In this case, this is what's called immunoglobulin M, IgM. And we will get to the different types of antibodies here in a little while. But you can see that these immunoglobulin molecules, these Ig molecules, this is a monomer configuration. I'll show you the difference between monomers, dimers, pentamers. But this is sort of the convenient architecture that we typically draw for an antibody. Okay, we usually just draw this monomer situation, which looks like a Y. And we'll get into more specifics. We'll kind of detail that Y out a little bit. I'll draw it a little bit darker, so maybe on the video you can see it. Okay. But I have a slide that's a little bit more obvious that's coming here in a second. But what I want you to take home from this is, we get the antigen binding to the IgM, and that triggers signal number 1. But in a similar fashion, we've got a CD molecule known as CD21 that's in the phospholipid bilayer of what cell? That's the B cell. And that CD21 is what interacts with the antigen. Look at this molecule. Do you guys remember this? Through a molecule known as complement. Do you guys remember the complement cascade? So here is an example of where this system of inflammation through the complement system is now actually helping to facilitate what's happening over here on our specific or adaptive immune system, because complement is facilitating or complementing the second signal of co-stimulation. Same kind of drill. Why do you do this? Well, what happens if something just brushes up and binds here that has specificity to this antibody, but it's not supposed to bind? It's a molecule that is part of you, it's nothing foreign, and it binds. Well, it also needs to bind to CD21 and have complement being released and available in order to send that second signal of co-stimulation. Okay, so let's draw this out. Right, so this is a B cell, and the secondary molecule, let's go to green, is our CD21. Right? And we have the antigen, and then we need a red marker for binding here. This triggers signal 1, and then complement triggers signal 2, and this is co-stimulation of a B cell. Guys follow? Question?
So if a bacteria releases a toxin that disrupts the complement cascade or any of the complement proteins, you don't activate the B cells?
Yeah, so the question is, what happens if a bacteria interferes or destroys complement? Would you have co-stimulation? Would you be activating B cells? In that situation, you wouldn't. Okay? There's lots of complement. It's blood circulating. So you might have a slower response because maybe it's kind of turning off or eliminating a population of complement, but there'll be more that comes in. But that's a very good question. When you ask those kinds of questions, it means you're thinking through like, oh, how does this work and what would turn it off? And if I was a bacteria and I wanted to find a way in, how would I do it, right? So this whole thing, this whole setup is kind of like a big war. But it's like guerrilla warfare, right? There's no real rules of engagement. And so the war is we have a defense system, like a military, which is our immune system, and bacteria and viruses are foreign invaders that are trying to breach into us as an organism. And so now we've got these different lines of defense. It's a great way of looking at it. I've explained it that way in the past. I've seen others explain it that way, and I think it's an example that's easy for us to understand our day and age. Okay, questions on that? So not many slides because hopefully you're taking some more notes here, and I think all of this is showing up pretty well on the video. Hi. That was weird. So my eye got really big. So you can kind of walk through this again. Right? But now we're going to dive into our next area of detail about what are these guys, these lymphocytes. So I want to explain, lymphocytes are not leukocytes. Leukocytes are these guys, white blood cells, neutrophils, monocytes become macrophages. Lymphocytes do circulate through the lymph system. They use the lymphatic tissue, and we're going to talk about that. That's kind of where they get their name. They're considered cells that really come from primary lymphoid organs or lymphoid tissue, bone marrow and thymus. And that's why we characterize bone marrow and thymus, and for sake of ease, we've labeled them bone marrow B cells and thymus T cells, so it's easy for you to remember. But the cells do get released into the bloodstream, and then of course they circulate around. You want your soldiers out there making rounds, okay, doing a perimeter check. And that's exactly how they behave. And then they report back in to a command center, which is considered a lymph organ. One of the reasons, how many of you, gotten a cold and then all of a sudden, like, the side of your neck or under here is like really tender? Okay, sometimes you're like, and this is embarrassing, but you're like, man, under my armpit kind of hurts. Has anybody ever had like a tender armpit? Okay, or how about behind the ear right here? Or how many of you had your tonsils removed? Okay. Okay? How many of you, this one's a little more awkward, how many of you kind of, it hurts on the groin and maybe on one side or the other when you get sick? Okay? All of those locations you have lymphatic tissue. You have lymph glands. Okay? And a big one is the tonsils that a lot of you raise your hand on. And so if you get an infection where the soldiers brought the infection back to the tonsils and then the infection wins, then you have tonsillitis and they just cut, the solution is let's just cut out we lost that battle, just take it out, right? You just nuke it, okay? Not like nuclear medicine, like you surgically remove it, sorry, I misspoke, okay? So you guys kind of get the idea. This picture up here is showing the thymus. So where is the thymus? Well, here's a picture of the thymus. The thymus is a little piece of tissue, okay, that's a pinkish gray color. It's soft, it's lobulated, and it's located just above the heart. Okay, right about where my lapel mic is. Okay, right basically below the knot in my tie. That's where your thymus is. It's about 5 centimeters in length and about 4 centimeters in width. It's about 6 millimeters thick when you're born. It enlarges during childhood, and then at puberty it starts to shrink or atrophy. Because what it's doing as you're a child is it's educating your immune system. The thymus is educating T cells. This is where T cells become immunocompetent. Okay, they become knowledgeable about what they're going to do when they grow up. At its height, or its peak, it sounds like we're talking about a ruler. It's 20 to 30 grams by the time of puberty, maybe upwards of 40 grams. And then in older patients, like at 75 years of age, you can't even find it on autopsy. It's like indistinguishable from the surrounding connective tissue, just kind of disappears. Okay, so the thymus goes up during childhood, peaks at puberty, and starts coming down. So your thymi, right? I don't know, is that the plural of thymuses? Thymus? Multiple thymus in this room? All of y'all's thymi? I don't know. So your thymus is on its way down, okay? And part of the reason is because your immune system is now educated. And so some of you anecdotally can attest to this. How many of you were pretty sick kids, but now you hardly ever get a cold? Okay? Yeah, so way to educate, right? You're reaping the benefit, your thymus reap the benefit. Now you heard this, I let my kids eat dirt because, it doesn't kill them, it makes them stronger, right? Well, actually, believe it or not, there's some truth to that Mississippi thinking, okay? I don't know if it came from Mississippi, but some of you are from Mississippi, I apologize, but, trying to be funny. Well, here's another one. How do you know that the toothbrush was invented in Mississippi? It'd be called the teeth brush if it was anywhere else, yeah, exactly. All right, so that one was horrible too. That's why my kids said, Dad, don't try to make jokes. Okay, so that's the thymus. That's where T cells become immunologically tolerant, or they become educated. But we use the word tolerant. Why do we say tolerant? So I want to talk about tolerance. Right? What does this word mean to you? Tolerance.
It's like you have a decreased reaction.
Yeah. Doesn't phase you. Right? You're not going to overreact on something. Well, that second part is really good. So you want T cells to be tolerant, meaning you don't want them to overreact to anything that is you. And so they're going to become educated and exposed to different antigens that belong to your body tissue, your cells. And we select for T cells that aren't self-reactive. So tolerant means not self-reactive. Self-reactive would be bad. A self-reactive T cell might seek out and decide to destroy maybe certain cells in the pancreas. For example, beta islet cells that make insulin. Right? And that's type 1 diabetes. I told you we weren't going to get into diseases until after the exam, but I want to give you, HIV, give you, diabetes, just to give you a snapshot of where all this is going. Why do we need to understand this stuff? Because then when we do, and you start seeing the etiology of the disease for type 1 diabetes, then you start to realize, oh, I get this. Well, I had self-reactive cells that actually destroyed that particular tissue. Okay? Same thing for multiple sclerosis. Okay? So we'll talk more about these examples after the exam, but I want to give you a couple of teasers so that you stay with me, so that you're not just because these slides are super busy, intense, and I get it. A lot of mechanism in the immune system. And sometimes showing you where we're headed and how this is going to relate and how you're going to be able to connect the dots is helpful. Okay? So, let's take a look at this busy graph. I've been like playing all on the margin, and you're like, what about all this crap? Right? Okay, so here's our thymus. Well, precursor T cells, whether they're B cells or T cells, precursor T cells come from the bone marrow. So the bone marrow is where B cells are, kind of maturing. But T cells ultimately come from the B marrow. They're called the bone marrow. They're called pro T cells, and then they go to the thymus where they become tolerant, not self-reactive, and they become immunocompetent. This happens right here in the thymus. And we've got this opportunity of self-reactive clones through a process known as central tolerance to be eliminated or screened. So in central tolerance, and I'm going to my notes here to make sure I don't misspeak. Okay. Central tolerance is happening in the T cells, and then out in the periphery, we actually refer to this, periphery just means outside of the thymus, as peripheral tolerance. So again, we have two backup systems. We decide centrally in the thymus, if they're self-reactive, we're going to get rid of them. So here we go, a self-reactive clone, right? Here's thymic epithelium. It sees the MHC complex. It reacts and triggers the cell for apoptosis. So I don't know if you remember in the very beginning of the semester we're talking about necrosis and apoptosis. I gave an example of apoptosis, digits of the hands and feet. Apoptosis normally is also used here to screen out self-reactive clones. So what we do in this process of the immune system is we spend a lot of biological energy coming up with different variations of how we're going to respond to antigens. And in the effort to create all the different permutations of what we might see in our lifetime and make T cells that are going to react, inevitably we're going to make self-reactive ones. So what we do is we signal them for apoptosis, programmed cell death, and we recycle the parts and we try again. Okay? For T cells, 95% approximately of the T cells that you make are signaled for apoptosis. Only 5% what you come up with actually makes it out to the system. That's how much biologic energy your immune system uses in order to see how creative can I be. And as you're growing up as a kid and you're exposed to more stuff in, first grade, kindergarten, preschool, first grade, all the way through, you get a lot of runny noses. You get a lot of kids sneezing on you, right? You get a lot of kids coughing all over you. You see it happen if you're a school teacher. One kid gets sick, and the next day, like 3 of them are sick. I mean, it spreads like a plague, right? If you know any school teachers. So, maybe mom and dad are. So we spend a lot of time here figuring out how to make all these creative T cells that are going to go police what's out there, but inevitably we create ones that are self-reactive, and so we signal them for apoptosis. The non-self-reactive clones, they just get out into the periphery, into the immune system, and they find a non-self peptide, right, bind, and then they have a T cell response, and they go do their thing. This is the normal pathway, okay? Now, if you've got a self-reactive clone that doesn't get detected in the thymus, and it makes it out into the periphery, we actually have 3 backup pathways to get rid of them. Let me say that again. If we have a self-reactive clone in the thymus, and it sneaks out, it leaks out of the thymus is actually the terminology that we use, and it's out in the periphery, now we use peripheral tolerance mechanisms to get rid of them. And there are 3. So number 1 is, bless you, is if you see that this is a tissue cell or an antigen-presenting cell that is showing a self-peptide, self-reactive, and here's a T cell, but look, here's that CD28 that doesn't have its co-stimulation, and so this guy is signaled for what we call anergy. The word anergy, The word anergy is a word that kind of sounds like anemic. It's a word that means the cell is rendered non-responsive. So remember when we talked about this co-stimulation of CD28? You don't have it, you don't get signal number 2, and the cell gets signal for anergy. Don't do anything. Must have been a false alarm. Make sense? Option number 2, let's go this way. Option number 2, we've got self-peptide, we've got our co-stimulation event, but this is self-reactive, so then we do activation-induced cell death, because in this particular situation, the activity is too aggressive. It stimulates it too frequently. So it's hyperactive, and because it's hyperactive, it's in an activation-induced cell death signal for apoptosis. So it's kind of like this Goldilocks syndrome. Too little, too much, doesn't work. It needs to be right down the middle and just right. Okay? Now our last pathway, so these guys are repeatedly being stimulated. Our last pathway that we kind of skipped through is down the middle. This one's interesting because this one you've got co-stimulation, but in this situation with overactivation, we trigger what's called a regulatory T cell to come in and suppress the response. So suppressor T cells or regulatory T cells are going to suppress the response. They dampen the response. They dampen the response by releasing certain cytokines and growth factors like interleukin 10 and TGF beta. And what they do is they turn off those pathways of activation. And so it's a non-apoptosis way to handle an overreactive, self-reactive T cell. Okay. Questions on this slide? Yes?
Why wouldn't you want to use apoptosis and the other two peripheral tolerance? Like, why would you want to be like triggered for anergy? Like, what's it gonna do since it's like binding area is gonna be matched up for like self recognition?
So why have 3?
Yeah, like like why keep those ones alive? Why not kill them with apoptosis?
Why keep these guys alive? I don't know the answer to that. The reason that's uniquely different is this one never got the co-stimulation signal.
Yeah.
And so it just goes into this cyclical pattern where it's kind of non-functional. And then in this one over here, these two really are more similar. And the difference here is this one never really sees the regulatory T cell. Maybe the regulatory T cells are not abundant enough, so they don't make it there in time. And so this process, by overstimulating, causes a trigger of apoptosis. If this regulatory T cell never showed up, then this is what would result. Okay. But I don't really have a good answer for your first question, which is a fair question. You know, why not just use apoptosis here? I don't know.
Do self-reactive clones only react with like our bodies themselves, or do they go like bacteria too?
Self-react. So the question is,
Maybe non-reactive and self-reactive.
yeah. Yeah, so the question is, good question. If you have a self-reactive clone, is it only reacting to self, or could it react to other things? It could react to non-self, but the problem is it does react to self, and so that's when it would be presenting issues, is when it might work just fine for a period of time, and then it starts seeing your own tissue, and then you have the onset of an immune disorder. So it's one of the reasons that some of these immune disorders lay quote unquote dormant, or they kind of don't show up right away, is this leakiness of self-reactivity can transition over time, right? So you can have self-reactive clones early on, but maybe they're not causing quite as much damage to the pancreas, or the beta islet cells of the pancreas, and then later in life, as the patient gets older, they start, growing a population that are self-reactive, and then usually they have some sort of episode, and I'll talk about this later, but something triggers them, and they get diagnosed as being diabetic. And so it used to be called, type 1 diabetes used to be called childhood onset of diabetes, but now you got patients your age, well out of childhood, that are being diagnosed with type 1 diabetes, And everyone's scratching their head going, I thought, I mean, if they were born with this, why is it showing up 22, 25 years later? Well, sometimes that self-reactivity takes time before it builds up or the whole organ gets compromised.
Does the immune, is it just an influx of like self-reactive clones, or is there something else inhibiting the apoptosis?
I don't know. I have a feeling that these things are functional, and maybe over time they become less capable of handling an increase in population of self-reactive clones. But again, I'm speculating on that one. Now I'm way outside of my area of expertise.
Is there some sort of chemical or like different protein that these self-reactive T cells have that is recognizable by these APCs? Because I'd imagine if they bind to a non-self-reactive, they're not gonna like overstimulate it or anything. They know what it is. So there has to be like a distinction, right, chemically?
Yeah, so the overstimulation is typically because the cell peptide is everywhere, right? So this is only showing one MHC. Like our drawings are just isolating one location, but what about over here, here, here? What about all 360 degrees? But remember, that's a 2D representation. This is a sphere. So if you have an entire baseball or basketball that's inserted with all these CD molecules, I mean, this thing could be lit up like a Christmas tree. Does that make sense? So you could have multiple of these events happening all along the surface of this sphere cell, right? It's a three-dimensional sphere. And so that's where the overreactivity is happening, like all over. Yes, sir?
So the overreactivity is the releasing of the chemokine? Is that what?
The overreactivity?
Yeah.
The overreactivity is you've got the cell getting stimulated into the nucleus of signal 1 and signal 2. It's happening repetitively over and over and over and over again, like nonstop. So imagine this is happening all over the cell, right? It's like a security alarm system in your home, but now it's telling you like 5 windows are being broken in and 3 doors all at the same time, you know, and there's a fire, you know, on floor number 2, you're like, what the, right? So the cell just is like, I'm out, yeah. Okay.
Just real quick, I just wondered, are all the CDs and all those markers, the MHAs and all that, are they mostly proteins?
Yeah, they're mostly proteinaceous, yeah. So they're amphiphilic, because they're sitting in the membrane. So they're gonna have fats, sugars, as well as protein associated with them, but the bulk of it is a transmembrane protein. I mean, this is all back to like basic bio, right? If you Google like a transmembrane protein, you're gonna have fatty sequences, right? Because they're amphiphilic, so they have hydrophilic and hydrophobic domains. And again, the thing that's cool about this class is, you know, we just said, oh, you just insert it in the membrane. But if you think back into basic bio of what is needed in order for a protein to be stuck inside of the phospholipid bilayer, and it's pretty complex. Okay. All right, folks, we do have to start going a little bit. So we are literally like on slide number 5 or something like that for today, which is fine. It's fine. We're going to take our pace as it comes. So at maturation, these cells reside in these secondary lymph organs, like in the tonsils, in lymph nodes throughout the body, in tissues, okay? And the activation is what we refer to as clonal selection. And so I talked about these clones. I alluded to these ideas of clones being not self-reactive. But clonal selection is a process whereby the immune system is able to create very creative variations of how the lymphocyte is going to interact with a potential invader. And so this is this idea, this clonal selection idea of we need to have variety and then we need to screen. So we have variations of clones. Some are non-reactive, some are self-reactive. Or, sorry, non-self-reactive, some are self-reactive. And so we screen out via central and peripheral tolerance for T cells which ones are self-reactive, and we get rid of them. Okay? And the ones that survive that whole process, like 5% of that population, is now circulating in the body in our lymphatics, hanging out in the lymph nodes until somebody shows up that you don't want there. Okay. These lymphocytes, kind of going back to this. So we looked at this diagram before. This is a CD4 positive cell, right? So this is a T cell. Here's our MHC complex 2. That's this diagram over here. We've got our co-stimulation event. We talked about our B cells. And so these cells are becoming activated in a couple of different ways. So the T cell receptors and the B cell receptors. The T cell receptors are this family that is receiving this antigen, interacting with the MHC class 2, as well as fashioning to the co-stimulation event in the antigen presenting cell, like CD80 and CD86. So our T cell receptors that you would write down would be molecules like CD4, our T cell receptor heterodimer with the alpha and the beta subunit, as well as our CD28 molecule. And then over here on our B cell receptors, for sake of completeness, right, we've got our B cell receptors that are immunoglobulin alpha beta subunit, and these guys are linked to our immunoglobulin that's a transmembrane domain that's binding our antigen. And then, of course, our CD21, which sees the complement as well as the antigen to give us signal number 2. And so our B cell receptor characters would be our IgA and B, our IgM, as well as our CD21. And these lymphocytes ultimately are activated or triggered by the presence of an antigen, right? And that antigen could be a bacteria, could be a virus. You don't want it to be a part of you. If it is, then you've got an immune disorder. We'll talk about that. Because what you do is you go out and you start attacking your own tissue. That happens. Okay. More detail. Just do a time check here. Okay, we're actually about where I thought we would be. Maybe a little behind, but we're okay. All right, so here's another schematic that's diagramming all this stuff that's happening on the board. More artistic, because I'm not an artist, I'm horrible at it, but you kind of hopefully you get the idea. So this now is a secondary lymphoid tissue. This is a lymph node or a lymph organ. And we've got antigen presenting cells that are going to show up. And these antigen presenting cells could be one of 3 characters. They could be B cells, macrophages, or dendritic cells. B cells, macrophages, or dendritic cells. So the B cell itself can actually serve as an APC, antigen presenting cell. The macrophage from over here, the tissue macrophage, can serve as an antigen presenting cell. And additionally to that are dendritic cells. So what are dendritic cells? So these dendritic cells kind of hang out at more epithelial interfaces. And they're labeled dendritic cells because they look like, morphologically, they look like a dendrite, like a neuron's dendrite. But they have nothing to do with the neuron. That's just their name. So the most confusing name for students is the dendritic cell because they think that I'm talking about something related to neurology or the nervous system. It's because they're very long and branching, okay? So they have a dendritic-looking shape, but they have nothing to do with the dendrite. In the skin, there's a specialized dendritic cell called a Langerhans cell. It's a type of dendritic cell. You might remember that from Bio 201. Langerhans cell. So in the skin, the Langerhans dendritic cell hangs out in the skin because that's your physical barrier, right? So if it makes it through the physical barrier, you've got antigen-presenting cells, the dendritic Langerhans cells, grabbing it, chewing it up, and now going to a lymphatic tissue and saying, hey, look at what I found in his left arm. Okay? So antigen-presenting cells. These cells can present to either a CD4 or a CD8. So they can either present to a helper T cell or a cytotoxic T cell. And you can see where they're going to do this. They're going to do this in the lymph system. They're going to do it in the lymph tissue. And so here is a dendritic cell with an antigen coming into a lymphoid organ, bless you. And you can see if it goes down this pathway of presenting, here it is showing, right? This is the dendritic cells displaying this antigen that it found in my left arm to a CD4 positive T cell, which is this 60% variety, which is the helper T cell. And it's saying, look at what I got. And now these CD4 positive effector T cells coalesce and they get activated. They move into the circulation, into the bloodstream, and they start migrating out, looking for that antigen marker anywhere in the body. When they find it, right, here's a CD4 positive T cell that's extravasated out, right? It's migrated out, very similar to the way that neutrophils and macrophages leave the vasculature, and you've seen that process now, and you know that process. And as it interacts with that same marker in the circulation, it can trigger macrophages to come in and destroy that cell, or it can release cytokines to trigger inflammation, which recruits more macrophages, okay? And then they gobble it up, they go to the lymphatics display as an antigen-presenting cell, and then the cycle continues until all of these cells that have this original marker that it found have been eliminated. Make sense? So that's how our helper T cell works. Aha, but that's only 60%. There's another 40% that can see that same marker. Let's go this direction with it. So now you've got the CD8 positive T cell, okay? It's displaying, we've got the antigen presenting cell that's displaying to the CD8 positive T cell, which is now our cytotoxic T cell, which is a 40% population in our body. These are approximate numbers. And again, similar types of things happen. They kind of bunch up, they become activated, and these are cytotoxic T lymphocytes. That's what the CTL stands for. I should have said over here, Th1 cells are a type of CD4 T cell that's now activated. So you see the Th1, that tells you, oh, that's a helper T cell that's pissed off. Okay? So Th1s are pissed off, activated CD4 positive helper T cells. The CTLs, the cytotoxic T cells, these guys are like BA, man. These guys, they get angry, they go into the circulation, okay? And here, this is this pathway over here. You can see, I gotta come back up here and explain that in a second. But once they get stimulated, they actually go and they physically attack whatever cell has that antigen on its surface. And that's why it's called a cytotoxic T lymphocyte, because this guy is actually carrying out the physical cytotoxic activity. The helper T cell uses a macrophage. It helps facilitate the response, but the cytotoxic T lymphocyte actually pulls the trigger, okay? All right, so let's back up to right here. So between the presentation and activation, we need to look at this blow up right here. And this is T cell production and differentiation. And so what's happening within that lymph tissue is a secondary co-stimulation event that triggers memory. So we've got our naive T cell. Here's our antigen presenting cell showing what it's found. And you've got the cell itself through an autocrine signal. It releases interleukin 2. It binds to itself, interleukin 2 receptor, and that creates this activation of cytotoxic T lymphocytes, as well as the creation of the production of memory T cells for the future. So if you ever run into that virus again, Now, you've already got these guys hanging around in the lymph tissue, and they can actually be recruited and released. Okay? So interleukin-1 through a autocrine, auto meaning self, we talked about that term before. It releases that molecule, it binds to itself on its own receptor. It's like throwing the ball up and catching it in the other hand. And that signal right there says, okay, let's make memory cells and let's go ahead and activate all of this population of T cells that are nearby me. All right, so let's look a little deeper at APCs, antigen presenting cells. So the left slide is a B cell with a bound peptide. The B cell is operating as a antigen presenting cell, one of the three. So you see the B cell is on the left side of the left picture, right? This guy right here. And here's our MHC with our peptide, and it's displaying it to a helper T cell because it's CD4. You know this is a helper T cell because it's CD4. Up top, you can see in the upper right, that's a macrophage. That's a macrophage, a cartoon diagram of a macrophage. That's these guys right here that we studied last week before. You got the bacterium being engulfed, right? It breaks it down, it displays the antigen on the surface. Macrophage is an antigen presenting cell. Here's our dendritic cell. You see how long and squiggly these guys are? So a Langerhans cell, same example, same type of thing. Here's our lymphocyte. This is a scanning electron micrograph. This is a pretty cool picture. Scanning electron micrograph picture of the dendritic cell displaying the antigen to the lymphocyte. Question?
So what happens to the APCs after they're finished presenting whatever antigens they have on them? Do they go back and keep doing that, or are they broken down?
So what happens to the antigen presenting cells after they present the antigen? Okay, so typically those antigen presenting cells will display their antigen and then they'll detach and they'll be recycled. Okay, so they break down through apoptotic mechanisms and then everything is kind of rebooted for them. All right, so we talked about this concept. We talked about what molecules present to what. I like this picture because it helps characterize that these MHC complexes that you see right here are not just a box diagram, right? So you can actually find online some of these video three-dimensional renderings of MHC class 1 and MHC class 2, and that's what this is, and it actually will rotate. I just took a snapshot of it to put it in the slide, but that would actually rotate so that you could see where the binding sites are, okay? And so there's a three-dimensional structure, but the MHC1 binds to CD8 positive, right? So CD8 positive is MHC1, so we should probably write that down. And then MHC2 is CD4 positive. And that nomenclature stays. That nomenclature stays. Questions?
So the dendritic cell has both MHC.
Say that one more time, the first part I heard for quick.
The dendritic cells?
Yeah.
They have both of these proteins in there.
Yeah, that's correct.
Okay.
Yeah. All right, we're going to shift gears on Monday, and this is where we're going to pick it up. What I would really like you to do is I would like you to take a look, obviously you're previewing the slide, the different types of antibodies, and then there's a drawing here, because I couldn't find a great drawing years ago, so I just made my own. I'm a horrible artist, but this took me like 7 hours, I think, to make this picture. I'm kidding. Okay? But I want you to be able to see and appreciate that this drawing is a representation of this monomer that's in this format right here. So here's our IgA monomer. Here's a dimer where there's two stuck together, right? Here's a pentamer, okay? So take a look at these slides, but walk through, be familiar with the forms or the shapes of antibodies and what their features are, so we can get through that on Monday. And what I'd like to be able to do on Monday, if we can get through the rest of this, is just this lecture, is we have a little time for some Q&A about the exam. Fair? So if we can go relatively efficiently on Monday and finish out this lecture slide deck, that's it for Monday. Okay, we don't have that many more slides, and we'll use the remaining time for those who want to chat a little bit about the exam. Okay? All right. Have a good weekend.